Hello, my name is Bill Webb, and I used to teach at Oaks High School. Some of you may have known me as Mr. Webb in the physics classroom. Others may have known me as Coach Webb on the basketball court. I currently am living up in the state of Washington, teaching at Cascade Christian School, but I still have fond memories of my time at Oaks, and once a lion, always a lion, so I've still got my Oaks gear here, and looking forward to sharing a little bit in this video about my experience with coding a couple summers ago at UCSB through a research project. So Shane Crawford is one of my OC Online students in AP Physics 1, and he asked if I'd put together this video to focus a little bit more on the coding side of what I did in that research project. So without further ado, let me show you some of the things that I learned and used in terms of coding for that research regarding COVID. So I guess it was just last spring. Some of you may have heard my presentation there when I was teaching at Oaks about this research, but in that lesson I was really focusing in on the biology and the chemistry side of things. And today I want to talk a little bit more about how I used the coding and what I was able to accomplish using that coding. But as I said, this was a research project at UCSB. It's called RET, and it was in 2023 when I started the project. It was through two different research labs, the Scott Shell Lab and the Joan Emma Shea Lab at UCSB. And there was some funding to provide the opportunity for me to come as a high school teacher during the summer and do this research. So I joined a team that was actually studying initially different kinds of proteins associated with Alzheimer's disease, but they were inviting me to join a kind of a branching team that was just starting that summer to investigate the impact of COVID and how hydrophobicity, or the repulsion of water, might have impacted the different variants of COVID being more deadly or more infectious. And so that was kind of the big picture of the research project that I was a part of. I'm not going to go through all the biology and chemistry. Some of you would have heard it last year, and even if you didn't, that's not the focus today, but I need to give you a little bit of background so you can understand some of the data we were crunching. So the SARS virus is what you see here in the green and the orange, and it attaches to these receptor cells on the surface of our cells when it infects us. Those receptor cells are called ACE2 receptors. And there's, as you probably know, a lot of different variants of the COVID virus. And so what we were trying to do is both visualize and analyze the hydrophobicity, or the repulsion of water by the various spike proteins on the SARS virus that would actually be attaching to those ACE receptors on the human cells. So this is an image made in a program called PyMol, and this on the left is just a ball and I guess it's kind of almost like a stick and ball or three-dimensional macromolecule picture of the SARS-CoV spike protein. And then on the right, we have more of the ribbon form of the ACE2 receptor protein that's on the surface of our human cells. And this is just a visualization so we could see which part of the ACE2 receptor actually came in close proximity to which part of the spike protein, so we would know which amino acids in those two proteins were actually binding together for the infection to take place. Now, anytime proteins come together, they have to push the water out of the way, because in the human body, all of this happens in an aqueous environment. So as you see here in the picture on the left, you see the two proteins bound together. On the right, they're pulled apart, and the portion that's colored kind of in that pink or purple color is the part that actually has to bind together with hydrogen bonding to hold those proteins together. And so in order for them to come close enough together for that binding to occur, it has to push the water of the environment out of the way. And so our theory was, and not just for this COVID research, but for lots of research happening at UCSB, is that possibly proteins that are able to push away the water a little bit better would be able to bind. And in the case of COVID, if they're better binding, then they might be more potentially dangerous or deadly, or they might just become more infectious. So it kind of depends on what that binding would lead to. If it would lead to a more complete infection, more cells being infected, more infectious in terms of being easily spread from person to person, all of those things could be factors. Now this is just a quick slide to show you all the different prototypes and variants that we were looking at. So the prototype is the original COVID virus, and these are just some of the variants we were working on. We mainly did our research with Alpha, Beta, Gamma, Delta, and Omicron, but there are lots of other variants out there, as some of you may know. I like this slide because it shows you in the RBD, which is the receptor binding domain, the part of the binding site where they connect, that there weren't really very many mutations when you go from the original COVID to the alpha. There was just one amino acid that changed. And then going from alpha to beta, there were just two other amino acids that changed. And so all these little red numbers and letters represent the amino acids and which ones were different from one variant to the next. So you can see when it gets down to Omicron, there were quite a few differences in the amino acids of Omicron versus a lot of the other variants. And so we were trying to analyze in this receptor binding domain, what was the hydrophobicity in this area of the protein for the different types of COVID, and especially around these amino acids where there was change, what did that hydrophobicity look like? And was it possible that hydrophobicity played a role? And so our questions or our objectives, would hydrophobicity and understanding the repulsion of water by the different COVID variants help us understand possibly why Delta variant was more deadly than many of the others, or why Omicron was much more easily spread amongst humans? It might even help us understand why different variants were better at escaping our immune system and infecting us, and even maybe predict what the next mutations could be, or at least the more dangerous mutations could be. So to understand how the hydrophobicity works, just give you a little quick background on water. When you have pure water, the molecules tend to form in these tetrahedral shapes where we have a very predictable 109.5 degree angle between any two different water molecules. And so what we were doing is trying to analyze 3 water molecules at a time and measuring those angles. So if we look at, as you can see in the side view over here on the left or the top view over here on the right, we're looking at a triplet of water molecules and seeing if that angle stays at 109.5 roughly, as it would in pure water, or if it fluctuates as that water starts to come in close proximity to a portion of the protein. So this is a little example of, we've got water molecules, just the oxygens of those water molecules represented by the red and the pink, and then we've got the green protein strand. I think this one's actually from one of the proteins that is being investigated for Parkinson's, actually. But just to help you understand, because there was a lot of different groups working on a lot of different research. But as we know, in pure water, it's about 109 degrees. But what we were doing is using software to actually measure the water molecule angles close to the protein. And so this triplet might have a 118.7 angle. This triplet might have an 85.3, and down here 144, and over here a 51.8, and over here an 86.3. And obviously we can't take a protractor and measure these molecules. They're way too small. So we have to use simulations to try to predict it. And so what we were doing is trying to have these computer simulations give us those angles, what they would do based on the natural repulsions and attractions of the water molecules and the protein strands, and see if we could interpret that data. So my workflow involved writing lots and lots of code. And I took some of that code and put it into that same program that we looked at the pictures before from, called PyMol, and generate a movie. So PyMol works all using Python. And so I wrote code to use in the PyMol program so that we could visualize the interaction of the water molecules with those proteins and then have it collect data. But the PyMol program itself can't collect the data effectively. So we had to write a different Python script and send it to a supercomputer pod cluster on the UCSB campus and have it crunch all the data, store all the numbers for us. So that was kind of the first part of my workflow, is to create that movie, do the simulation, send the results of the simulation to the pod cluster, and get back the data from it. So here is the movie that was created with some of the coding that we wrote. And so all the little red and white dots are the water molecules, and the green in the middle is the spike protein from COVID. And you can see the water molecules are constantly vibrating, and even the protein is changing its shape and its position within the water. And the software was designed to help measure the triplet angles, like I showed you on the previous slide, of all the water molecules 3 layers deep all around the protein. So again, as you notice in this video as it plays, not only are the water molecules moving, but that protein is shifting its position, shifting the angles of the different branches on the protein. And the software could measure the angles, those triplet angles that I showed you, for millions of different molecules, just a few layers deep all the way around the protein. And so here's a picture of that pod computer cluster that we were using. It's in this giant room. It's all highly air conditioned to keep everything running well. And the band of computers we were using are these 4 over here that have all the dolphins across the top. And so this bundle of computers was the cluster we were using. And depending on whether I was working from home or whether I was working from campus, I used a variety of different software interfaces to be able to send my code to the pod and have it do the work for us. Now afterwards, it would send back data frames of information, and I wrote another Python script to take that data and then to compile it, organize it, and then push it out into CSV files, or basically Excel type files, so that we could more easily analyze it. So had to write scripting and code for making the movie, for sending the batch files, for sending it to the pod cluster, more scripting to take that data frame information and convert it to CSV files. So there's a lot of coding. And this is just a few snapshots of some of the code and some of the results. So over here on the far left, all this numbers along the screen in the white background, that's what the data would come back from the pod cluster as. And then you can see the kind of Excel looking CSV file that showed, okay, this is a particular amino acid, and it gives us all the information we need to be able to predict the hydrophobicity or hydrophilicity of that portion of the protein, amino acid by amino acid. And over here on the right top, you see with the blue lettering, that's the batch file, or at least part of the file that was used to send those batches of information to the pod cluster, so it would send me back the information. And then down here in the middle, this is one of the softwares that I used or created to then take that information and put it into CSV files and then organize it. And then over here on the far right in the bottom, this is how I would take that information from those CSV files and use it to color the pictures that I'll show you both on graphs and in some of the models so that we could visualize it better. And there were a lot of other codes that I wrote. I don't even remember how many different coding programs that I had to put together, how many scripts I put together. Probably around a dozen, I think, different scripts to do different functions. Some are really short, some are very involved. But I learned a lot about coding, and I learned a lot about the effectiveness of using it, and I did different things with it that I'd never done before. So one of the things that we did was take that data and then have it be graphed. This graph wasn't created on Excel or any other software. It was created using Python. And so we would have it look at the data and tell with the script to create this graph with all the correct angles, all the correct labels, the key over here. Everything was done using Python, and you just had to write up the script to make it generate this graph. And that's one thing that I learned is at the high school level, all the graphing that we usually teach and do is using things like Desmos or graphical analysis or the TI Inspire software or Excel. But I didn't really realize you could just use Python scripting to do that. And if you have some good AI resources like Microsoft Copilot, you can, I think, or maybe it's not Microsoft, I can't remember. The Copilot software that I used was a great AI for helping me generate code. Or if I was getting stuck, it could kind of help me find the problems that I had in my coding. I wasn't an experienced coder when I went into this research, but spent hours and hours and hours every day for 6 weeks learning to do it better. And a lot of artificial intelligence resources were helpful for me to learn it well, to kind of fact check and spot check the problems that I had so that I could generate graphs like this. These are some other graphs we generated with the Python, and we could even focus in on, okay, well, here are the amino acids that were different from, this particular ancestral version of COVID to the Delta version. Those are the two within the receptor binding domain that were different. And we could measure what is that signature in the hydrophilicity of it and in the hydrophobicity of it and try to compare that. This was another one where we converted that hydrophilicity and hydrophobicity data and had, again, Python do some calculations with that data and predict what's called de-wetting free energy. And that is how much energy does it cost or take to de-wet the surface of the protein, to remove the water from the surface of the protein so that they can bind like I talked about before. And so this was a different measure, and again, showing the different mutations, and specifically the mutations in green that were near the areas of the protein that actually were involved in the binding connections. So we could try to see, was there a difference? In this case, it was comparing SARS and Omicron. Here's another one where we were looking at alpha, beta, gamma, and the original SARS all together. And we're, again, using Python to write the code, going amino acid by amino acid, measuring out the de-wetting energy. And so just all of this is, like I said, something that I was not aware of, and making graphs using Python. So learning to write those scripts was a fun challenge. After we got all those data and made the graphs, then we would take the data from those graphs after it had been organized in a variety of different ways and put it back into the PyMol software using, again, another script and have it generate models. So basically, all the data that's in these graphs that I showed you, plus here's another one up in the top right-hand corner that I hadn't showed you. So there was even more than what I've shown that we used the Python coding to help us generate the graphs, and analyze the data. We would take all of that and then organize it with another script to send it back to the PyMol software and generate these pictures. So in this picture, everywhere where it's bright red, that is very hydrophobic. And everywhere it's blue, it's very hydrophilic. And white is kind of right in between. And then the black that you see there kind of in the middle, so it's a black background, but you can see some of those black spheres kind of in the middle in this opening. Those are core molecules that are kind of embedded internal to the protein that wouldn't have much access to the surrounding water. And so we colored them black if the software simulation told us that there were fewer than 10 measured angles on those amino acids. And so we can see, if you're familiar with amino acids, you can see some of the shapes. So this pink one right here, if you can see the mouse kind of moving around, and this dark red one up here, those are particular amino acids, and you can see by their shape. So we were having it analyze the angles amino acid by amino acid within this protein. So some of the advantages of using computational analysis along with wet lab research, because while the group I was with was entirely focused on the computational side and the computer side of it, there were several other groups doing wet lab research, more traditional test tubes and beakers and centrifuges and things like that. But some of the advantages of the computer analysis that we were doing is we had far less disposable and consumable costs, because we didn't have to have all that glassware, and we didn't have to have all the cleaning supplies, and we didn't have to have all the sterilization techniques. And so there's a reduced cost in that regard. There's also a reduced cost in terms of efficiency and time. Most of the labs that the wet lab group were doing would take weeks, if not months, before they could get data. And using the pod computer cluster, I could send information, and even with that really high-powered computer, it would sometimes take a couple hours to get the data back. But I could get that data back in hours instead of weeks or months. And so there was a time efficiency as well. And then because we weren't working with actual viruses or other harmful or potentially harmful biologicals, we didn't have the legal or safety regulations and risks that they have in those wet labs. And so that also was a definite advantage. And then one of the advantages I see for students like yourself that are learning coding is that you are developing marketable skills. So the better you can do coding, the more opportunities you're going to have as you go forward, even in the areas of scientific research. And I would really strongly encourage you, for those that are planning to pursue a scientific study at the next level, as soon as you get into college, I would encourage you to, in your departments, wherever you're studying, find out if there's research projects you can get involved in. Because if you have some coding background from high school, they're going to be really excited to draw you in and involve you in the research. Sometimes there's some advantages financially for you if you are able to get into some of those activities as well. So for yourself, I think it helps develop those skills that could be very marketable if you're looking to get into specific colleges or to involve yourself in research once you're there. So I hope that's helpful for you. Thanks to Shane for inviting me to be involved. I wish I could come in person, and we tried to figure out if there's some way I could maybe Zoom live so we could have a Q&A, but the time you're meeting is right at a time that I'm actually teaching physics, so at my new school. But miss all of you at Oaks, so for those of you that I know, I'm glad to kind of connect with you, even not in person, and hopefully this video has been helpful in some way. For those that I don't know, again, I hope this was helpful for you. If you do have any questions, perhaps Shane can put you in connection with me. I still do actually have my Oaks email address because I'm teaching part-time for OC Online, so you can also reach out that way if you have questions. So thanks again for inviting me, and I hope you have a great day.